So I hope you can see my screen. And today I'm going to talk about module 4, that is nucleic acids. And I think some of you have already read, done some quiz. Some people have done some quizzes. So feel free to ask questions if you felt something was unclear. Okay, it's not a problem. I don't mind being interrupted at all. So what we have learned in previous modules, and things are a little bit moved a little bit forward just because of the exam. We have the schedule is a little bit shifted. So we have learned about lipids, and now we are heading to learn towards nucleic acids. What are they made up of? What is their function? What do they translate into? And what are the biological consequences of radiation? We know that radiation directly affects the DNA. So it's important that we cover that kind of information while we are talking about nucleic acids. Since many of you have done Bio 101, 102, or other biology classes, you might already know that nucleic acids are, or commonly known as DNA and RNA, are important molecules, and they are important in the sense that they store genetic information. And that genetic information gets passed on from one generation to another generation. So their primary function is to store the genetic information, and they're also responsible for passing this on. What is the monomer for nucleic acids? So every time we talk about these macromolecules, whether it is carbohydrates, whether it is lipids, whether it is nucleic acids or proteins, we always talk about what is monomer and what is the polymer. So for nucleic acids, the monomer is nucleotide. And these nucleotides, one of the nucleotides is shown here. It is comprised of 3 different parts. We'll come to that. But for now, it's important to know that they are connected to form the entire structure, which we call nucleic acids. And that is a polymer. So nucleotides are the monomer. That is what we are starting from. And these repeated units, they are connected by this, what you see the P here. So P is the Phosphodiester Bond. Okay. So this means that there is a covalent bond which connects them from one monomer to the other monomer. So just let's take a look into the structure of the nucleic acids, which is really important. The nucleotide is comprised of 3 different things. And what often happens is that when you are remembering this, sometimes you may learn this now, but then later you may have to come back to it because it essentially is 3 totally different things. So you may have to come back to it to remind yourself what it is. So let's start with something which we are most familiar with. So we have studied about carbohydrates. We have studied a lot about hexose because we were talking about glucose or galactose. But in this case, the sugar here is pentose. Okay. So when we are talking about pentose, we mean it's a 5-carbon sugar. So just to make sure that you are able to see the 5 carbons, I'm going to write the carbon here. So this is C1. Sorry, this is, that's C1. So this corner, that's C2. And actually it is called prime. So C3. This one is C4. And finally, this is C5, the one that already has a C. The reason why I'm showing this to you is during the oral exam, I observed that a lot of people, when asked for anomeric carbon, were very tempted to just mark the C that is shown. Please don't forget that there is a C which is present at every corner of the structure. And that is as much as a carbon as this one is. So this one is the C1 prime, 2 prime, 3 prime, 4 prime, 5 prime. And you may say, why are you numbering them? You will soon see the reason why I'm numbering them, and that this knowledge will help you in the next few slides. So the first carbon is connected to a base, and the last carbon is connected through a phosphate bond. It is connected, and this is what, if you see in the last slide, is what takes you to make the bond between the two monomers. Okay, so this is the carbons here, and this is forming there is a sugar in here. The name of this sugar is ribose. Okay, so I'm just going to name here. And the ribose is a pentose sugar. What is attached to it is a purine or pyrimidine base. We will soon see what do we mean by that. For your reference, what is shown here is a purine base, which is a double ring structure, and a pyrimidine, which is a single ring structure. And the last part is the phosphate bond. So the ones that are highlighted in green are the 3 parts. That is also number 1, which is corresponding to the entire structure that is shown here. However, if you are just asked what is nucleoside, then you do not count the phosphate bond in there. You're just going to count this part. Okay. And if someone is asking you about nitrogenous base, you are only going to be mentioning purine or pyrimidine base. Okay. So I hope that it is clear that these 3 things are referring to, one is just to the base, one is to base with sugar, which is nucleoside, and the last one is base with sugar with phosphate, and that is nucleotide. So that is the difference between nucleotide and nucleoside. If you have any questions about that, I can repeat this information again.
I'm a little confused with it. Can you go back and explain it again? I'm sorry.
Of course, yeah, it's not a problem. So let's go this time the reverse way. We are going to start with number 3, or from the bottom. So first of all, there is a base. Why is it called a nitrogenous base? Because it has nitrogen on its structure. So that is why we call it nitrogenous base, because obviously carbon is going to be there. Okay, but this one has nitrogen also. So nitrogenous base can be of 2 types. One is pyrimidine, which is long name but only 1 ring. And the other one, funny enough, it has a short name, which is purine, but it has double ring. So that's how I remember it. I always remind myself, the one with a short name has 2 rings, the one with a long name has 1 ring. So that's just the base. When you combine base with sugar, which means that they're actually forming a bond, that is going to be nucleoside. And when you have base and sugar with phosphate group, that whole thing together, this combined thing is a nucleotide. We haven't talked yet about ribose or deoxyribose. We haven't yet talked about that, but we are soon going to do that. Okay. So does anyone have more questions from here? And please feel free to share what specific question it is, or it could be even be like, I think this may be hard to remember, or it could be like, I don't understand what this base entails. So just ask your question without any hesitation. Okay, we can come back to this. So what is a base? We kept talking about this nitrogenous base or nucleobase or bases in a nucleotide. What is a base? So let's just talk only about the base. Okay. So the base, it is a ring-like structure, and you can see when there are these alternate double bonds, then we call them, then these have become aromatic. By aromatic, I mean like, they have this distinct smell, aroma, so aromatic, okay? Why they are heteroaromatic? Because they have these different molecules in them, different, atoms, different types of elements in here, okay? Now about purine, it has more, I guess it has two rings. One is a hexose, which means like, sorry, not hexase, which is a hex ring, which is six. There is 6 carbon and nitrogen combination, and one of them is a carbon nitrogen, but it's a 5 member ring. Okay, so planar, or these are planar structure that is there on one plane. So you can imagine this screen is a one plane or two-dimensional plane. That's how these are. They absorb UV light around 250 to 270 nanometer. So you may think, why is this information all of a sudden given to me? Am I supposed to remember this? So for anyone who is trying to do any kind of DNA assays, any kind of biochemical assays, or if you're going to study more of biochemistry or going to study more of biological sciences and DNA, this is a very important property of DNA and RNA, that they absorb light around 250 to 270. So even their purity can be measured by this, or they can be detected at this range. So this is an important fact, and that is why it is mentioned over here. Now, when we talk about, so this is about bases, let's talk about sugars. There is something really interesting about sugars. Let's, without looking at all the text, focus on the figures. So there is ribose and deoxyribose. Can someone, by looking just at the figure, tell me, why is deoxyribose called deoxy?
Because it doesn't have the oxygen.
Great. Yes, absolutely. So it does not have the oxygen. So we are going to focus only in this blue part. Ribose has the oxygen, regular ribose. This is devoid of the oxygen, and so this makes it deoxyribose. Now let us think about the full name of DNA and RNA. So DNA is called deoxyribose nucleic acid, and the RNA is called ribonucleic acid. Okay. So these are the two, like the full forms, and these are the nucleic acids that we are going to be concerned about. These are the most important molecules inside a cell. And we're going to talk more, but it's important for you to recognize early on that the name of DNA and RNA is based on the sugar. When the sugar has two OH bonds, then it is called then it is called ribose. When there's one of the O is missing, it's deoxygenated, that is, oxygen is removed. Okay, so like degrade or deteriorate, so it is gone, then we call it deoxyribose. And you may say, does this oxygen really make a difference? It makes a lot of difference. Okay, it gives them very different properties, which we are going to learn as we go through this module. So nucleic acids are linear polymers consisting of sugar, phosphates, and bases. Yes, we know that because we were actually looking at that in one of our previous slides. We know that they are polymers containing sugar, bases, and phosphate. So we know that part. There are two types of nucleic acids, DNA and RNA. Now we know through this slide that the DNA contains the deoxyribose sugar and RNA contain the ribose sugar. So when you're talking about the DNA and RNA, then the DNA provides the code for cellular activity. Okay, so what does that mean? So that means that there is genetic information that is to be stored in a cell, and it has to be preserved in the right form to be passed on from generation to generation, and that's what DNA does. And what does RNA does? RNA helps convert that code which is preserved in DNA into proteins. So both of them perform really important functions. So let's take a good look at each of them individually, because in the previous slide, we were doing a comparison. Now we are going to focus only on deoxyribonucleotide. So deoxyribonucleotide will have a deoxyribose sugar, a base, and a phosphate. So you may ask, what is this base? So we're going to get to that soon. This base can be either purine, which is a double ring structure, or pyrimidine. You might have heard of adenine, guanine, cytosine, and thymine. A, T, and G pairs with C. So we typically call them ATGC. We are going to talk about the pairing in a minute. But if you look at this purine, there is you're not expected to remember the structure. Only thing you're expected to remember is purine is adenine and guanine. Pyrimidine is cytosine and thymine. So how do I remember this? So I found a trick to remember this, because sometimes it will get confusing. Which ones are purines? Which one is pyrimidine? So pyrimidine has a Y in it. Let me do it a different color. So I'm going to do a green. So if you see here, pyrimidine has a Y in it, cytosine has a Y in it, thymine has a Y in it. So I'm never going to be confused. I know whenever someone says pyrimidine, it is the one with the Ys. Does adenine have a Y? No. Does guanine have a Y? No. Okay. So we kind of keep the Y stuff together. There is, however, an exception. When we will talk about uracil, that is also a pyrimidine. We will talk about it in the next slides. Okay. So we are not going to rush to that. But thymine and uracil are almost similar in structure. So uracil and thymine will kind of go together. Thymine goes in DNA and uracil goes in RNA. Okay. So adenine and guanine are double ringed purines, and cytosine and thymine are smaller single ringed pyrimidines. And that is how the nucleotide gets its name. We are just relying on the base to give the nucleotide its name, because otherwise the sugar, which is deoxyribose, and the phosphate, it's all the same. Differences created because of the bases. So again, going to what difference is created, if you can see here, different bases can be attached here. You can have adenine here in this part here in the cream colored section. You can have guanine here, you can have cytosine here, or you can have thymine here as far as the DNA is concerned. And please note that doesn't have the OH, it just has the H. So what does DNA form? DNA forms then a double helix structure. So now we are putting all of this information together, and let's just focus on the right side. So I'm going to just block it for a minute so that you don't feel like there's too much information in the slide. So if you see here, then you will find here that this is a thymine. So that's the base, sugar, phosphate. Similarly, this is the guanine. These belong to the same strand. So let me repeat what belongs to the same strand. Okay. These belong to the same strand because as you can see, they are connected here. This connection is called the phosphodiester bond. Let me write that down for you. Phosphodiester bond. Now for those, if you remember your module 1, you will remember that an ester bond looks like this. It's literally an O molecule, which is combined to different C's, okay? So there can be a C here, and that's your ester bond. That's your ester molecule, right? Now, in this case, O is combined to phosphate and carbon. O is combined with phosphate and carbon. And there are two ester bonds, ester bond 1, ester bond 2. So that is why it's called a phosphodiester bond, which connects each strand. So you can see this is not connected here, not connected by sugar, only connected at this end. So that is one strand of the DNA. Okay. And let us look at the other strand now. Very similarly, there is base here, sugar here, and phosphate here, and it is connected to the other end. Okay. So now I have a question for you. My question to you is, do you see that these two strands, which is the one strand which is circled in red and the other one which I'm circling in green, do you think that these two strands, they are connected to each other?
They're connected on the, what is it, the 3' end?
They're connected here, of course. Are you talking about this one?
The one that you circled in green.
Okay. So you are saying they are connected over here. Okay. So let me just remove these because, you know, all of these can get a little bit confusing, and still hide this one here. See if I can. Okay. So I'm going to hide this here. I'm going to now bring your attention to just this part. Okay. So let me see if I can do a better job with this. So let's just focus on this part. Do you see my highlighted green part? That is the connection between the two strands. So imagine my one hand is one strand and one hand is the other strand. And what I'm showing to you is that connection between my two hands with the help of this pen. And so this is actually the hydrogen bond. Okay. So where is the hydrogen bond? It is present between oxygen and hydrogen. And in this case, it is present between nitrogen and hydrogen. Okay. Similarly, I can see there is hydrogen bond, which is present here. These are the two that are bringing the two strands together. Otherwise, these strands were actually connected by phosphodiester bond. So essentially, I'm trying to bring your attention that the phosphodiester bond keeps the strands together, each of the strand, making each of the strand is done by a phosphodiester bond. But when the strands interact with each other, that is with a hydrogen bond. So those are combining the bases. Okay, so this is an interaction between the two bases. So I want to stop here and ask if you have any questions about what I just said to you. Okay, no questions yet. Well, you can ask me if you feel, because sometimes this information can seem a little bit, it takes a little bit time to digest. Also, please note that adenine is connected to thymine and guanine is connected to cytosine. Okay, so also let me ask you this question. How many hydrogen bonds are between A to T? How many hydrogen bonds do you see? Similarly, my question about GC.
Five.
Just between AT, how many are there? It's actually shown here with this black lines.
Two.
Two. And how many in the GC? How many do you see? That one is not labeled, but I'm pretty sure you can count and tell me.
Three.
Three. So another question for you to think about is, which one is easier to break? Is AT easier to break or GC easier to break? The one with more bonds is easier to break, or the ones with less bond is easier to break?
With less bonds.
Yeah, so the AT is easier to break. And you may say, why do we have to break them? Well, when there is DNA replication happening, these are getting broken because the strands are separated. Okay, so that's what I have to tell you, that AT is easier to break because GC has 3 hydrogen bonds. Let me show you here. Hydrogen bond 1, 2, and 3. And this has 2 hydrogen bond, that is hydrogen bond 1, hydrogen bond 2. Now you may say hydrogen bond, is it always only found between oxygen and hydrogen or nitrogen and hydrogen? Yes, it's pretty much formed between either hydrogen-hydrogen, oxygen-hydrogen, or nitrogen-hydrogen. It's very specific kind of bond. Hydrogen has to be involved and it has to be bonding with specifically with these. So that is the hydrogen bonds. And that is, towards the end is the phosphodiester bond. Now one more thing to bring to your attention, count the number of carbon, 1, 2, 3, 4, 5. So that is why this end is called 5 prime and this end is called 3 prime because it's ending in 3 on this side. Let me color it in a different color for you. So note that this is 5 prime and this one is in 3 prime. And in the other side, it's 3 first and 5 later. Okay. So what does this indicate? So I'm going to show my hands on, I cannot see my window because I kind of closed it just for the ease of explaining. But you can see that if I put my hands like this, it's not parallel, it's anti-parallel. That means they're parallel, but they are heading in different directions. Notice the sugar. The sugar in here is looking up like this. In this case, the sugar is looking down like this. So these are anti-parallel structure. One of them always goes from 5 prime to 3 prime. So this one is going like this, and this one is going like this. So I hope you appreciate that these are strands that are running opposite to each other. With this, now I am ready to show you the picture on the left side of this slide, where adenine is shown by red color, thymine with yellow, guanine with blue, and cytosine with green. So you will always see adenine and thymine red and yellow together. You will always see guanine and cytosine, that is green and blue together. And do you see how they are parallel to each other? They look parallel, but they are actually anti-parallel. One of the strands runs like that, and the other runs like from bottom to top. Are there any other questions from this particular slide that you might have for me? Okay, nothing else. Okay, so these are the base pairs. This is called the sugar phosphate bonds. So now my question to you is, on the structure on the left, can you tell me where is hydrogen bond present and where is phosphodiester bond present? I'm just labeling the hydrogen bond with green so I can, once you tell me the answer, I can label it there. So where should I put the hydrogen bond? Is it towards the inside or towards the outside? Does anyone, you can take a guess. I mean, outside is which one? The
Hydrogen.
So you're saying outside is the hydrogen. Okay. And then the phosphodiester is inside. Now, Good. Thank you for responding because it kind of helps me explain further. So look at the base pairs where A and T meet. That's where the hydrogen bond is. Where are A and T or G and C meeting? They're meeting over here, right? Sorry. Do you see that they're meeting over here? The green and sorry, the red and yellow, or green and blue, they're meeting over here. So where they are meeting is where the hydrogen bond is. Now, where is the phosphate bond? Do you see the outside? So I'm just trying to show you how similar these two structures are. So where they are, where it says sugar phosphate backbone, that is where the phosphodiester bonds are present. Okay, so inside is the hydrogen bond and outside is the sugar phosphate bond. So my question to you, next question to you is, why is DNA negatively charged? And the answer is within this figure. That is why I mean, there is a clue in here. You should be able to tell why is DNA negatively charged. Because of what? Is it because of the base or the sugar or the phosphate? Is there a problem in understanding this? Or is that, you can just put it in the chat. I mean, you can still guess, or you can just send it to me. That's fine. You should still try to answer because it will help you. These are some of the questions that can easily come in the exam. That's fine. Okay, I see here one response, the phosphate group. Well, Aisha, you are 100% correct. The phosphate group. And look at the phosphate group. Let me clear this for a second. Do you see the negative charge in here? The negative on the ends? These are the negative charge here. So these are all indicating that the DNA is negatively charged because of the phosphate bonds, the presence of oxygen and phosphorus, and that creation of negative charge in here. That's why. So these ends are all negatively charged. You know, you see this negative charge running on the strands. Okay. So now that we have spent enough time on this, we can come back to this, but let's get back to RNA. RNA has a very similar structure, except that it has a ribose sugar and it also has bases, just like DNA. Is there a difference in bases? Yes. You have to remember an important difference in bases. You can see that in bold shown here is uracil. We had seen that purines are guanine and adenine. In DNA, pyrimidines were cytosine and thymine. In this case, thymine is replaced by a uracil that is found only in RNA. So whenever we talk about RNA, we always say that A pairs with U and G pairs with C. So G pairs with C remains the same, but A, instead of pairing with T, it pairs with U. Okay. So RNA is single-stranded. Just to show you, RNA is single-stranded. So you may say, what is the huge big difference between DNA and RNA? So you can see there are now many differences between DNA and RNA. And RNA is quite unstable. Just that presence of one extra oxygen makes it unstable. And so it is important to notice the difference in that structure. So essentially, these are all the purines and pyrimidines. We have now combined them together for your benefit to see, to show to you that purines are adenine and guanine, and pyrimidines are cytosine, thymine, and uracil. Thymine being present in DNA and uracil being present in RNA. So let's look at the big picture. DNA is found in all living organisms. Of course, there is exceptions like viruses. Eukaryotic cells have a central controlling organelle called nucleus in the center. There are chromosomes which are present inside the nucleus. These chromosomes may vary from different organisms. And then chromosomes are, entire DNA of each cell is condensed in the chromosome. These chromosomes, if you were to expand them, then they will expand. Like if you see here, this is a chromosome. If you were going to untangle it and expand it, you will see that there is DNA strand in here. And this, so that means that DNA is the one that is combining with, you know, these circles, they're called histones. They combine together and they form these chromosomes, okay? And these chromosomes are a part of nucleus. So essentially the information is stored here, and then it is actually packed in the chromosome inside a cell. So if you talk about DNA, one important question is, we often use the terminology haploid, diploid. What does it mean? Haploid means one copy of chromosome, diploid means two copies of chromosome, and polyploid means several copies of chromosomes, okay? So in human beings, you already probably know that one set of chromosome means 23 chromosomes. So the diploid number is 46. If I give you a reverse question, can you tell me the answer? If the diploid number in an organism is 50, what is the haploid number?
25.
Okay, excellent. So that is 25. That's great. Now you may also question me, like, who has polyploid? So many plants have polyploid. For example, strawberry. Strawberry has only 7 chromosomes. Okay, so if you say the haploid number is 7. Okay, now each chromosome has 8 copies present. So polyploid. So I'm talking about still about strawberry. It is polyploid, means it has 8N. When we were talking about diploid, we were saying it is 2N. Okay? With polyploid, we are saying it's 8N. So that means that every strawberry has 7 times 8, which is 56 total chromosomes. It's pretty amazing. It's totally amazing to see. The how many number of chromosomes are there. Okay, so you have to remember just a very simple thing. Haploid means one copy, diploid means 2 copy, and polyploid means many. In human beings, one set of chromosome comes from mother side, and the one set of chromosome comes from the father side. That is why every individual has 2 copies of chromosome. In plants, the division is a little bit different. Now let's talk briefly about RNA, because we talked about DNA and its position, like what is its role and what is its function in the cell, where is it located. Similarly, we are going to talk about RNA, and all of this sometimes can constitute a single course. Like there are people who would just study about DNA, RNA, translation, transcription. We are here going to focus only on very specific things because it's a 2-credit class. So I'm trying to give you information that we can manage to do in spending the right amount of time. So there are 3 different types of RNA. One is called the messenger RNA. Second one is the ribosomal RNA. And third one is called transfer RNA. So mRNA transcribes the genetic code from DNA to proteins. So mRNA is extremely important. It is, once the DNA is converted to mRNA, we know that mRNA is going to be converted to protein. So mRNA carries this information from nucleus to the cytoplasm. So if you remember, nucleus is that central organelle which controls the activities and functions of a cell. So it goes out of nucleus like a messenger and brings it to the cytoplasm so that proteins can be made in the cytoplasm. The second one is ribosomal RNA. So here is the picture of messenger RNA. What is ribosomal RNA? Ribosomal RNA is present in the cytoplasm of the cell where ribosomes are present. It's right here. And it helps in the translation of messenger RNA. So I'm not only telling you about ribosomal RNA, I'm also telling you how it is connected to the messenger RNA. The last, but still equally important, is transfer RNA, which is located in the cytoplasm. Transfer RNA brings amino acid to ribosomes, okay? And, I know it's kind of difficult to imagine why it would do that. We are going to take a look at translation, then we will understand. But it brings the amino acid, and these amino acids are the ones that are bound together to form protein. So transfer RNA is fetching the amino acid from cytoplasm, bringing it to the ribosomes, and helping the messenger RNA to be converted to protein. So for you, it is important to know that these three forms of RNA do an important task. Comparison of DNA and RNA. We know that DNA and RNA are having a little bit of structural difference, but when we put them together, this is something that this is an important slide because it's showing how DNA is converted to RNA and RNA is converted to protein. Actually, this should be more like not just comparison, but flow of information. So DNA is converted to RNA, specifically messenger RNA. This process is called transcription. Messenger RNA is then converted to protein. This green part is the protein, the yellow are the ribosomes, and this red is the messenger RNA. So if you see here, this red messenger RNA is converted to protein with the help of a process called translation. So essentially, this slide is important from two standpoints. First, it shows how DNA is converted to RNA, is converted to protein. Then it names the process as transcription and translation. And there is a very easy way to remember that. So I'm going to show you how you can remember that. So when you think of transcription, arrange transcription and translation alphabetically, and what comes first. So automatically transcription with the C comes first, translation with the L comes later. So that's why transcription happens. Not that's why, but it makes it easy to remember that transcription happens first and translation happens later. If you have any questions from this slide, you can ask me. Okay, how does this actually happen? Okay, so let's first focus on the left side, the definitions. Transcription converts DNA to RNA. Translation converts RNA to protein. Okay, how to remember? Arrange the words transcription and translation alphabetically. First there is transcription, then comes translation. Now let us come to the part how this happens. Now it's really, really important for you to focus here because this question is going to be in your assignment probably. It's going to be in your exam, going to be in the quiz, whatever that is. So it is extremely important that you pay attention and let me know if you don't understand something. So DNA to RNA, how does this happen? So this is the DNA strand, if you look here. Okay, and if you see here, you are, let me block off some of the part of it. Let's just focus on this part, okay? You have to remember that A pairs with T or U, I always write it like that, and G pairs with C. So it goes without saying that if you see a T or U, if you see a T, it pairs with A, and if you see a C, it pairs with G, okay? So now if you see here, T, if the DNA has T, RNA will have A. If the DNA has A, RNA will have U. So you can see here how we are using this complementary base pairs to put the complementary base on the RNA. TAC. Corresponding nucleotides are A, U, G. Similarly, the next one is T, corresponding is A, T, corresponding is A, corresponding to C is G, A is U, and so on and so forth. Don't yet look at the translation. We don't need that information. We're not going to worry about it right now. We will worry about it in a little bit. So we're not looking at this. We're just focusing on this. So I have a small exercise for you. And my exercise is like this. I'm going to write this here. And you can just, I'm going to write only 9 things, and you can just write to me what is the RNA sequence. And you can write to me, you can write to everyone, that is fine. And I will be telling you why it is first TAC, but let's first get done with this. TAC, and then the next one that I have for you is C space A space G, and the last one is G, T, A. This is the DNA sequence I have for you. So for a minute, don't look at this. Oops, sorry. Let me just cross it off. So for a minute, don't look at this, okay? And just look at what I gave you and convert it to oops. Why is it not making? That's interesting. It should have made. I can just make it disappear, or I can try. Okay, for some reason it's not working, but don't look at this pink one. You make your own. This is your DNA sequence. Write down what is the corresponding RNA sequence. Okay, someone texted me, someone chatted with me or sent it to me. Okay. Okay. Yeah, whoever just messaged, this was good. Yeah, I think many of you are getting it. Okay, I'll just wait for some more responses, and then I will explain it one more time, because this is important for you to know. And in my opinion, everyone should try it. If you don't try it now, you will have to do it anyway, later. So for your ease, I'm going to block it off in 3 threes. Now it is making, when I don't want it to make so big, now it's making. Okay, I'm going to block it off as in 3-3. Okay, and you will see why it is done. It is always done 3-3. Okay, so now that makes it easy. Okay, so now I'm going to write, and I'm going to try to write it in pink so that it will feel like it's RNA. So we just said that A pairs with T or U, T in DNA, but we are making RNA. So let's not get confused. A with U, G with C, T with A, and C with G. Okay. So now all we need to do is read the second part. So there is T here. So I'm going here. Okay. T with A. Okay. Let me write A here. Next one. A, I need to find out for A. Corresponding to A is U. Okay. I need to find for C. Corresponding to C is G. Okay. And I go here. Corresponding to C is G. For A, there is U, because we can never put a T. Okay. And with G, there is C. So if you can see here, I am just following the base pairing rules and putting them together. TAC becomes AUG, CAG becomes GUC, and finally GTA becomes CAU. If I went too fast, you can actually tell me And I'm going to block it off in 3 so you can actually see it much better. This corresponds with this with this. If this is unclear for any reason, you can please ask now. Anything that is not clear, you can ask now, because I'm pretty sure others might have same question.
So you can just never put T in only.
That was a great question, China. Yes, you cannot put T if you are making RNA. Now you may say, when will I even need to do DNA? Well, if you're doing DNA replication, you might be expected to, or if I gave you one strand and I told you, China, what is the other strand? Then you'll say, obviously the other strand is going to look just like DNA. So let me clear this up. If you see here, T pairs with A pairs with T. So yes, you do have a T here, but correspondingly you have a U in RNA. Similarly, keep going, everything looks same. A, T, 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 U, 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 U. Whatever is T in DNA is actually becoming a U in RNA. Okay. Any other questions about how we are doing this process of transcription? Okay, and then the pairing of threes. Why are we pairing in threes? Because this is 3 makes a codon. That is why we are pairing. And so we are going to talk more about it. I don't want to talk everything in one slide. I just want to keep it piece by piece information. And this is the process of translation. Okay. So this part essentially is like finishing all the important things that we needed to learn. In summary, DNA is double-stranded, RNA is single-stranded. DNA has a deoxyribose sugar, RNA has a ribose sugar. In DNA, the bases used are A, T, G, and C. However, in RNA, there are 3 common bases, but one of them, uracil, is different. These are the differences between DNA and RNA. The time is right around 3:30. Here are some of the radiotherapy slides, which are easier to understand as you read them. So I'm not going to focus too much on them. And you can listen to them in the other lectures because I have posted lectures. But this part just talks about the history of DNA and how 4 different people were involved in the discovery of DNA. And this particular slide talks about A is equal to T and C is equal to G. Who formed this rule? And that was Chargeff's rule. Erwin Chargeff found this rule. And he told that there is A plus, we all know that all the bases combined together, A plus T plus G plus C is equal to 100%. We know that the amount of A is same as T, amount of C is same as G, because they're always paired to each other. So this is the DNA base pairing rule. That is why if you see here, the AT and CG are always paired together. You can also see in this figure sugar and phosphate. And since we discussed, you should know that inside is the hydrogen bond and outside is the phosphodiester bond. This is essentially showing the same thing, just emphasizing on how it looks like, what are the distances between them, not that you have to remember, but it's really, some figures make more sense to some people and to others make some people, some other figures would make more sense, just showing you the exact same thing which we talked about for AT and GC base pairs. So these 2 chains, they are complementary because if we know one, we can know the other. They are also running anti-parallel because 5' to 3' is for one chain because of the number of carbon, 1, 2, 3, 4, 5. And 3 is like 1, 2, 3. So 3' to 5', 5' to 3' here and 3' to 5' here. Then we talk about gene. Gene is a single unit carrying information on a certain protein. In human beings, there are about 30,000 genes. So that is only 1 to 2% of the entire genome. The rest of the 98% of our own body's, the DNA, we do not know what it does. Okay, so that is the important information. The rest of the DNA we call junk DNA, and we don't think it's junk, but we don't know what function it performs. So here comes the part where we are going to talk a little bit about translation. So just to make sure that you are familiar, if you look at the figure in inset, it is the figure for transcription. Now we are going to talk very briefly about translation. For doing the translation, we need to know a term. The name of the term is codon. Codon is a sequence of 3 nucleotides that encodes for 1 amino acid. Okay. Let me check. Yes, so if you see here, this is the whole table, and you can see here that there is, on this side, there are different arrangements of the nucleic acids, and these arrangements can give rise to an amino acid, which means that if you take UGG, so for finding UGG, I say U is here, and then G is here. This is my UGG. Then I know that this is a tryptophan. So I write here TRP. The next one is UUU. How do I find that? I say this is U. There is U. So it can be found in this block. UUU is right here, and that is the phenylalanine, or which is PHE, or you can also write it F. So with the help of this codon table, we can actually translate the information in messenger RNA to protein. And you will always be given this table. All you need to do is take groups of 3, put them in here, and then let yourself see what is the amino acid that results in. So anyone has any questions about this? Is this something that you have done? You might have done. If you have not done, please feel free to ask. I'm happy to explain it all over again. It's not at all a problem.
Can you explain it one more time?
I'm happy to explain it because it is a difficult concept. So let us focus on this part. I hope you have understood the transcription. We know that in transcription, A pairs with U and G pairs with C. So based on that, we have generated the RNA. This RNA has now migrated into the cytoplasm. There is ribosomes here. If you remember, there is rough endoplasmic reticulum and smooth. Rough is containing ribosomes. Ribosomes is where this messenger RNAs will go. And that is where this conversion will happen. How will it happen? Because in messenger RNA, when we see the first 3 codon, UGG, what we do is and this in our cell, it happens. Our cell has that piece of information through transfer RNA. But if you look here, UGG, and I'm going to, let me make it a little bit thicker. So if this is U and this is G, then essentially the information that we need is in this block. So we're trying to find UGG and we say, okay, I found it. UGG is right here. What is the amino acid that corresponds to UGG? Tryptophan. So I can either write here W because there is a W below that. That's perfect. Or I can write trip. And I mean the first one, not the second one. I know this is in the middle somewhere. Now let's go to the next one, UUU. And for the UUU, we are going to hear U and then U. So it has to be in the first block itself. And now, okay, I found it, UUU. And that is phenylalanine, or you can also call it F. Now, next is GGC. So for the GGC, we go here, this is G, this is G, sorry, yeah, so G and G. So that is going to be, we are just going to take the cross column. So G and G, are in this place, you can also find it by yourself. You didn't really need to go through this process, just makes it very systematic. GGC, I found it here. This codes for a glycine or a G. So I can either write glycine or I can write G. Both are correct answers. Last one is UCA, which would be U is right here, C is right here. So it must be talking about the first block. Okay, I found the UCA. That's going to be this one, corresponding to serine, or you can write it S. So with the help of this table, which will always be provided to you, no one is going to expect you to pull this up or memorize this or anything. You will be able to make the structure of protein or construct the structure of protein. This conversion from RNA to protein is called as translation. If there is anything unclear in this, you can also ask me specific question like the second was not clear or third or the usage of this table. I want to tell you one more thing. I want you to focus on also on, I'm going to mark it in a different color. I want you to also focus on this thing called start code, methionine, which is a start codon. Oh, didn't end up marking it in different color. Okay, so AUG is the start codon. And look at this, if you find UGA, it is stop codon. So we also need something to start the process, and we need things to end the process. We don't want the protein to go on and on and on. So there are start and stop codons, which will actually start the process or end the process. So if you would have encountered UAG, instead of S, you could have written just stop. So just getting familiar with this table, that there are different amino acids, there are 20 different amino acids, and that's how they are. So this is not something that scientists have made. This is something that exists in the nature, and scientists have just deciphered it and put together in a table. Okay. So let me clear these drawings. Any other questions from this slide? We're almost getting done, actually. There's not a whole lot left. Okay. So you should definitely expect some question on this. And so this is for you to practice and try out by yourself. Transcribe this and translate this and try out by yourself. So I really want to encourage you to do this by yourself, try it out. And of course, you can share the answers with me. The only place where I hesitate to explain over the phone is when there are structures involved, and I don't want to, I want to make sure that you're not confused.
So if it doesn't have a start or stop codon written next to it, then it's not one or the other, right?
If it doesn't have a start in the beginning and a stop in the end, well, for your purposes, I think we are just doing for practice.
Right.
But in the real life scenario, there has to be a start and stop, because otherwise the bodies or the cells machinery will not know when to start and when to stop. So that's a great question, Paris. So the stop codon has to be there. Now, Paris, what Paris asked me brings us to an important question. Okay, I'm going to ask the question in a different way. Have you heard about the word mutation?
Mutation?
Yes.
Yes.
You might have heard it in different contexts. You might have even heard it in the sci-fi movies and all whatnot. Sometimes when these conversions or DNA replications are happening, things can go out of control in the sense like something can change. Like when this is getting converted, like for example, you know, I'm trying to see if I can, okay. So for some reason, if instead of this T, let me just cross it out. suppose by mistake, DNA puts an A, and instead of the G, there is a C. What will happen? So let's try this. A pairs with U, so there will be a U here. I'm going to try to bring it closer. T pairs with A, and then C pairs with G. So this single mutation can result in UAG codon. But there are two mutations here. Sometimes it's just one mutation. When that happens, and UAG, I know we do a process, but you can see here UAG is right here. So I know UAG is a stop codon. So what will happen is this was going to be a normal protein. Because of mutation, it became a stop codon. So your body cell is fooled now. Your body cell, instead of making this entire protein, which was going to do an important function in the cell, just started making the protein, and because of a mutation hit the stop. So the presence of stop codon in wrong places can result in a protein being shorter or a protein being longer. So which means it is very important. Those location of stop codons is as important as the location as each of these proteins.
So
Or, yes, go ahead.
what I'm trying to figure out is so arginine, right? Am I pronouncing that right? Arginine. It doesn't have either a stop or a start. So it's neither a stop or start. Is that correct?
You mean arginate, you mean like this one?
Yeah.
Argin-
Yes. The word stop or start isn't in there. So I'm just trying to figure out.
Oh, yeah. Don't worry about it. This is nothing to do with the location of these. What you have to remember is to first convert all of this. When you convert all of this, you will see that in this case also, you will end up with a stop codon in the end. So where should stop come? Stop should come always here. If I give you a sequence, usually you should expect that this should be the stop. And you may say, is this a stop in this case? Absolutely. A pairs with U, C pairs with G, and T pairs with A, and that is a UGA. And when you give a UGA, you know that a UGA is right here, which is also a stop.
Oh, okay. I see.
Okay. So any kind of change in nucleotide is a tricky business for the body, and that is also results in cancers. Okay. So it is very important that the DNA duplication as well as transcription happens accurately in our bodies, in all living organisms for that matter. So with this, we have finished this like very conceptual part, and I know it's taken a little bit of time. I do want to tell you that after this, we are just talking about how the DNA gets damaged. That's why I introduced the word mutation to you. Mutation is an alteration or a change in sequence, and it can happen due to many reasons. So this is something that you can read why it can happen because of many environmental factors. It can happen because of factors inside the body. And you will be surprised to know there are 10,000 mutations in every cell in every day. You know how many cells we have? We have about 100 trillion cells. Every cell can undergo mutation. And since it can go mutation, there is DNA repair machinery which fixes it. So you can imagine if we did not have a machinery, or if for someone the machinery is not working right, it can result in mutations that kind of stay in the cells. And you might have also heard that UV rays and ionizing radiations, these are they can introduce mutations inside the DNA because they can break or change the structure of DNA. So here is an example of that. Once there is UV light, it can make a dimer between the pyrimidines. Now you know that pyrimidine is not supposed to connect like that. They're not supposed to be bonding like that. There is a way they're supposed to bond. But if they just kind of bond like that within each strand, that's a bad thing. And that's why When you are, that's why they say when you're out in the sun and you didn't wear the sunscreen and you're out there for a prolonged time, your skin cells might undergo this kind of reaction where the thymine and thymine will combine together to form a dimer. And sometimes some of those damages get repaired, other times they don't really get repaired. So UV light can cause different kinds of reactions between DNA molecules. And you can see that's the dimer that's getting formed. So it forms a kink. It destroys the structure of the DNA. And when the structure of the DNA is not maintained, then that DNA will not be able to give the RNA in the right manner, and it will result in a mutated cell. Okay, so this is just the other part of it. What can destroy the DNA? First part was all understanding what is DNA. Can things destroy the DNA? Yes, they can. Ionizing radiations can destroy the DNA by directly hitting them like that, or through the water molecule. This can ionize the water molecule, which can hit the DNA. So there are so many different things that can result in this. And so this is how radiation therapy works. Radiation therapy works because we are introducing radiation to cancerous cells and letting them die. But does it affect normal cells? You must have often heard when you apply radiation, it can also affect the nearby cells and it kills them unnecessarily. Okay, so this is what this slide talks about. And then there is a little bit on, because you are in the allied health. So just talking a little bit about how does radiation therapy, how does it play an important role in treatment? And what are the different types of therapy which are present? And of this, the proton beam is very focused one and has least effect on the area of, what should I say, like the nearby cells. So there are certain advantages and those are kind of listed here. Also, the radiation is used for detection. And we are aware of these techniques because either us or someone else in the family would have gotten an X-ray or a scan done or a fluoroscopy done, and these are all based on radiation. So just bringing to you a little bit effect of, just the information on radiation through these slides. Okay. So I'm going to stop sharing. I'm sorry it took a little bit longer, but I do want to make sure that I'm explaining from my side. I'm going to stop recording now.